our lecture on the effects on microbial growth kinetics, we will now look at the effects of environmental conditions on the growth. So the first, of course, condition that is most often seen and considered, especially in the industry, is the temperature. So the growth here shows that here is the optimal temperatures, is the rate of growth versus a different types of temperature. Now, the optimal temperatures we can see here is at around 30 to 40 for most microorganisms. We call this one the mesophiles. Then for the thermophilic organisms, the thermophiles, they love higher temperatures. So the optimum growth rate is around 60. So here, as you can see, different microorganisms thrive on a variety of um, temperatures. And actually, we even have hyperthermophiles, where these are usually the archaea. They thrive on temperatures at around 90 to 100, so around near or around boiling points. And then going on the other end of the spectrum, we have the psychrotrophs and the psychrophiles. So psychrotrophs thrive on a lower temperatures, while psychrophiles, they can only thrive on lower temperatures. So here are your basically microorganisms. When we talk about the temperature, of course, we talk about their heats. So heat generation during microbial growth. So as we can see, since there is an optimum temperature at which your organism grows, if you want to maintain your fermentation at the optimum temperature, you need to consider the amount of heat being generated. So in here, around 40 to 50% of the energy from the substrate is converted to ATP for aerobic growth, and the rest is heat. So there is a maintenance requirement which is low for rapidly growing cells. So basically, your energy, it now boils down to where the energy comes what happens to the energy from your substrate, because at the end of the day, the ultimate source of this heat comes still from the substrate. So they are the ultimate source of heat and, of course, material. So the energy from the substrate is basically divided into the metabolic heat and the delta H of the cell material. So here, in terms of, if we represent them as a chemical equation, we have the metabolic heat 1 over YH plus the heat from the cell material is equal to the heat of the substrate. So the metabolic heat is basically, this is, well, it can be considered a constant. So here is a calculated metabolic heat for a typical bacterial cells over certain different substrate. So basically, you will now here observe that for certain substrate, there is some substrates produce greater metabolic heat compared to others. For example, glucose produce greater metabolic heat for the bacteria than, say, methane at 0.061. And this metabolic heat now is correlated to the rate of heat evolution. So going back to your energy balance, if you remember, Q is the amount of heat that you need to remove during fermentation. Otherwise, your fermentation will become overheated. So that heat comes from metabolic heat. So here is Q sub R. This is the heat generated for the growth. This is around, we can calculate them by getting the volume of the liquid V sub L in liters, and you have, of course, the net growth rate multiplied by the number of cells concentration times your metabolic heat. Or roughly, if you have aerobic or aerobic fermentations, you can roughly correlate here into the rate of oxygen uptake. So this is roughly around 0.12 of the heat generated for sorry, not heat generated, the heat generated in the by the metabolic metabolism is around 0.12 times the rate of oxygen uptake U sub O2. So because of, especially for aerobic organisms thriving on glucose, whereas you have a high metabolic heat, you now have there is now a necessity for getting a cooling water to regulate temperature in your fermenters. So here is your pH. So another consideration is pH. So here is just like for the temperature, there is now a rate or a spectrum at which some organisms thrive on certain pH. So you have the acidophiles, the neutrophiles, and the alkalinophiles. So the pH, in terms of pH, so if we look at temperature, the metabolic heat is, we can see it in terms of, say, oxygen uptake. We can roughly correlate it in terms of oxygen uptake. But in pH, these are affected in turn by the nitrogen source and the evolution and consumption of carbon dioxide, as well as if there are any acidic or basic byproduct. Of course, the more acidic byproduct you produce, the more acidic the culture media would be. So, in terms of nitrogen source, most of the nitrogen sources is basically ammonia. So ammonia is basic. So there's a decrease in pH. So you need to consider that. If it's a nitrate, so is it coming from nitric acid, which of course is acidic? And then if it's a salt, nitrate salt, you need to also consider the because nitric acid is considered a relatively weaker acid. You might want to consider what would be the effect of that on your pH. In addition to that, you have the carbon dioxide. When dissolved in water, it is carbonic acid. Of course, it's an acid, so it becomes acidic. Aside from that, we have lactic acid and some even alcohols. Then we also have, aside from the pH, dissolved oxygen. So dissolved oxygen is an important substrate in aerobic fermentation. This is often our limiting substrate because oxygen is sparingly soluble in water. The critical oxygen concentration, if you have a critical oxygen concentration, if your growth rate is above the critical growth concentration, the growth rate is dependent of the dissolved oxygen content. But below the critical oxygen concentration, your oxygen becomes now a rate limiting factor, of course, because it is a limiting substrate, becomes a limiting substrate. Now, there's an important consideration here that at high cell concentration, the rate of oxygen uptake by the cells might become limited because, of course, you need to consider diffusion, the diffusion of oxygens into the cell. And that is actually one reason why in some layouts of fermenters, oxygen, the air is sparged from the outside. It also serves as pressurized oxygen is sparged from the bottom. It serves not only to provide air and mixing, but also it allows for a faster diffusion of air in the fermenter. So here is another consideration. So growth rate is different from the extent of growth. When we talk about growth rate, it's limited by the dissolved oxygen, while the extent of growth, or how long will you maintain to be at exponential phase, this is controlled by the number of the amount of carbon source that you have, the amount of substrate that you basically have. Now, the typical value for saturation at saturated dissolved oxygen at 25 degrees Celsius is at 7 ppm. Now, the requirements for some bacteria and yeast, we have at around 5 to 10% of saturated DO. That is the critical. So, above the critical, the growth rate is not affected by DO, but below the critical, the oxygen is now a rate limiting factor. And then the molds, which depends on the pellet size. Again, there is an issue here of the diffusion of oxygen. That's around 10 to 50% of the saturated DO. Then we also have the last named factor, which is the redox potential. This is an important parameter that affects the rate and extent of many redox reactions. So in fermentation, affected by DO, pH, and ion concentrations. So because we have reducing and oxidizing agents here. So in order to regulate the redox potential in the fermenter, they introduce they pass through nitrogen gas, which is also a neutral element, and some reducing agent like cysteine or sodium sulfide. So they increase. If you want to increase the redox potential, you add oxygen or oxidizing agents. The reason why we need to monitor this one, because the redox potential, if you have high amount of becomes so high, you will now have reactive oxygen species in your fermenter, and that is not good. That is basically toxic to the cells. So other factors, we have dissolved carbon dioxide, which can be toxic, but sometimes in some metabolic functions, you might also need dissolved carbon dioxide. So you just need to regulate the carbon dioxide content of your air supply or your agitation speed. Also, ionic strength, which affects the transport of nutrients across the cell membrane. So you have high substrate concentration also can have inhibitory effects. In fact, if you read about production of secondary metabolites, you would notice here that, for example, in penicillin, there must be a limit in the amount of phosphates. The phosphate must be a limiting in order to properly produce a high amount of penicillin. So there are some cases like that. We need to intentionally limit some substrates or some micronutrients in order to achieve um, the highest possible yield that we want to have. Okay. Okay, so let's now move on in the same video lecture with quite long models to predict the specific growth rate. Here is there are models, and it is a plural. We are not limited. In normal microbiology, in your industrial microbiology, we might have had already discussed only one model, the microbial growth model, the exponential growth model, basically. But that is the most simple one. But it does not really accurately predict everything, especially in the actual practical result. Okay, and the reason are this. So there is some difficulty in calculating growth kinetics. So some compositions and biosynthesis change with regards to these environmental conditions, even if we want to maintain same concentration, same pressure, temperature and pH in the cell. As the cells grow, the composition changes. So there is now a variation between a balanced growth and the unbalanced growth. So it's easy to do it in balanced growth, but it's difficult to calculate and predict in unbalanced growth. So there's a need to recognize the structured nature of cell and the segregation of the culture into individual units, because they are not uh, each cell is unique, basically. So of course, in relation to that, we have models, structured models and unstructured models. When we say structured models, these are the models that consider the cell mass that consider the cell mass as different components. So they are proteins, carbohydrates, lipids, nucleic acids. So the structured models take into account the differences in the type of components of your cell. So versus the unstructured, where it is just summarized at a certain constant. They are counted only as one. Then we also have segregated versus unsegregated. Segregated is that each individual cells are different from each other. Unsegregated is we treat them as one unified system or body. Now, because the structured models and the segregated models treat the different components of the cells and the different individual cells as unique, of course, they are more computationally complex than the more simplified, unstructured, and unsegregated models. So here are some examples of unstructured, non-segregated models. So we have substrate limited growth, models with growth inhibitors, and logistic equations. So let's look at substrate limited growth. In substrate limited growth, the assumption here is the growth kinetics is dependent on a single substrate. We only consider substrate. For example, it's sugar. The amount of glucose is basically this is the limiting substrate, and the growth kinetics is dependent on this one. It follows your saturation kinetics and enzyme kinetics models of the Michaelis-Menten equation, which is obtained from the Monod equation. So basically here is your substrate limited growth. So your growth rate constant is equal to your mu m mu max times the substrate over Ks, where there is your Michaelis constant times the number of substrate that you have. So it's the substrate that is the one that's dictating how your organisms will grow. So it's basically again an application of the Michaelis-Menten equation. So, however, this is only applicable when the growth is slow and the population density of the cell is low. But however, in practical purposes, it's not always the case. At high population densities, there is a rapid consumption of substrate and accumulation of toxic metabolic waste. Therefore, there would be some variations now in your substrate limited growth model in order to account for your uh, basically the high consumption of the substrate and of course your metabolic toxicity. So here are some alternatives to this equation. We have the Blackman models. Here is the equation for Blackman models. Basically, in Blackman model, they fit this graph. So basically, all of these models are designed so as to fit the observable experimental data. So the Blackman model fit the graph better than your monod, but this one is a discontinuous model. So in test year has the advantage of having less calculation constants than the Moser model, which is full of many different constants, variation constants, and the Contour model has the saturation constants that can describe a substrate limited growth at high cell densities. So Contour basically answers the issue with the monod model here with high population densities. Now, how about models with certain growth inhibitors? For example, substrate inhibition. So, substrate inhibition can be competitive or non-competitive. So, basically, in these models, if you notice, they are actually considering the cells as the catalyst and they're applying catalyzed reaction kinetics with cell catalysis in these models. So, substrate inhibition, whereas your substrate there's a competitive inhibitor. The competitive inhibitor here becomes your growth inhibitors. So if the growth inhibitor can act as either a competitive inhibitor or an non-competitive inhibitor. So here are some models with your growth inhibitors. Actually, this is can be explained by the simple Michaelis-Menten model. So here is the normal enzyme, but there's a competitive inhibitor. You have a different Km but the same Vmax, and a non-competitive inhibitor with the same Michaelis constant and a different Vmax. In this model, this is basically a Michaelis-Menten model applied to um, the microbial growth model, provided you have a process of a growth inhibitors. So the growth inhibitors can be the product of the cell themselves. Product inhibition is observed in some cells. They can be competitive or non-competitive. So an example for that is, for example, in production of ethanol, fermentation of alcoholic beverages. There's only a certain number of alcohol. Actually, this has been a practical problem before because especially in the beer, there was an issue where a company wanted to increase the amount of alcohol content in the beer. And it's very difficult because the strain of the yeast that they are doing, the strain of the yeast cannot handle the high alcohol concentration. Even if they input here a large amount of sugar substrate, they the wort, they enrich the culture with a very high concentration of sugar, the alcohol content stays the same because the yeast cannot handle it. They cannot live on a very high alcohol concentration. They produce alcohol, yes, but that is also toxic to them. So there's actually a need to find another strain that has a greater alcohol tolerance. And then aside from that, there are also the production of the toxic compounds in the cells. Okay? So that's the models with the growth inhibitor, because it's actually an actual fact that the growth of the cells in a fermenter is inhibited by sometimes by the products or the accumulation of a toxic byproducts. Okay, and we now have another one, which is the logistic equation. The logistic equation, this is a very old, if you have heard of the logistic equation, it has a wide variety of application from biology to chemistry, and then even to economics and ecology. The logistic equation, it shows here that over a certain time period, there is an increase because, let's say here, the hallmark of the logistic equation is a sigmoidal or S, the letter S shaped curve. It's called a logistic because it follows only um, two functions, no, two conditions, or no, it's not a condition. It follows uh, two cases where there is a limited amount of population and there is a large amount of population. So in a limited amount of population, you will now observe an increasing growth rate, but it will come a time that it will reach what you call the carrying capacity of the environment. The environment can no longer support that amount of population, whereas the growth decreases until it becomes zero, or there is no longer a change in the population. It becomes stagnant. So this set of equations characterize growth in terms of the carrying capacity, basically the ability of the environment to accommodate that number of individuals. And this is actually seen in batch cultures also. Remember, we have a stationary and dead phase precisely because the environmental conditions can no longer support them. There's no longer substrate, the nutrient source is depleted, there's a lot of waste product. So that is seen in your logistic equation. Okay, so these are the models, some of the models for predicting and characterizing or describing our growth in a bacterial fermentation. So let's look at an example here for an ethanol production in batch. So fermentation of glucose to ethanol has been one of the most thoroughly studied process because a lot of the beverages, and even not only that, um, ethanol is not only an additive in food, but is also an important chemical for cleaning, and especially now after the pandemic, all of us has a bottle of alcohol in our pocket. Fermentation of glucose to ethanol is a process here. We have your glucose and other nutrients to produce the cells, the ethanol, and its byproducts. So it's a microaerobic operation. That means only a small amount of oxygen is needed because they are added for the sake of a fatty acid metabolism. And hence, you will observe here a Crabtree effect, whereas your oxygen is depleted once exponential growth phase occur and oxygen is purged by the vigorous application of carbon dioxide. We only need to limit the amount of oxygen. So product inhibition in the ethanol production is observed when you have an increase in ethanol concentration. The fraction of uninhibited ethanol productivity, these are actual experimental data, is seen to decline because of the high concentration of ethanol. Of course, it's basically somewhat like a feedback inhibition loop. And of course, the toxicity also of ethanol to the yeast. So here is a summary of the byproduct inhibition. So this is not only observed not only in ethanol, but also in other products. Whether you are a primary metabolite product or a secondary metabolite product, there is also an inhibition mechanism there. That is why it's considered one of the models in microbial growth kinetics, because inhibition is basically a real thing in batch fermentation. So you have this fumaric acid, acetic acid, lactic acids, you have acetaldehyde as well. So they are mostly chemical interference. And with this in mind, here is a process flow diagram for fermentation system for molasses. Taking into account, of course, you have the maintaining the certain optimum amount of temperature, pH condition, oxygen content, and the amount of metabolic waste that can be produced. So it looks a bit difficult because in real life, fermentations and manufacturing processes are really multi-step processes. Okay, and that's it for this lecture on microbial growth kinetics.